Good morning, good afternoon, good evening. Welcome everyone to today's Global IMMUNOTALK. Global IMMUNOTALKs are an important initiative created in 2020 to spread immunology and inspire the new generation of immunologists all around the world in a friendly and egalitarian manner. I am Gabriel Rabinovich and have the honor and privilege of presenting our today's speaker, Dr. Mauro Gaya. Mauro Gaya obtained his first degree in molecular and cellular biology from the University of Buenos Aires in 2011 and his PhD from the University College London. He performed his PhD work at the London Research Institute in the laboratory of Dr. Batista. While there, he uncovered a role for lymph node resident macrophages in the induction of B-cell responses upon infection, a publication published in Science in 2015. Then Mauro performed a short postdoctoral stage in between the Francis Crick Institute in London and the Reagan Institute of MIT and Harvard, still with Dr. Batista. His work led to the discovery of how innate T cell populations initiate the seeding of germinal center reactions during infection, a publication published in Cell. Mauro Gaya was recruited to the Lumini Center for Immunology in Marseille, France in 2019, where he studies B-cell immunity in barrier tissues. During this period, Dr. Gaya became a Marie Curie Fellow, tenure researcher at the National Institute of Health and Medical Researcher in CERN, and EMBO Young Investigator. At present, his lab is funded by an ERC starting grant program to study the role of IgA in B-cell memory. Mauro is also deeply involved in mentoring graduate students and actively engaging in extension activities, including organization of symposia and workshops. In a personal note, I would like to say that I am particularly touched about presenting Mauro today. I coordinate the immunology course at the School of Exact and Natural Sciences at the National University of Buenos Aires, and he was undoubtedly one of the best students I have ever had, and I am extremely proud of his stellar scientific career. But before you start your talk, Mauro, one of the traditions of Global IMMUNOTALKs is to ask our speakers a special question. The question for you today is, can you describe a turning point in your career that defined your current research focus?
Hello, Gabriel. Thank you very much for your kind presentation. And also thanks to you because when I was studying biology at the University of Buenos Aires, I was deciding if I was going to orient myself into studying plants or immunology. And in fact, during the same semester, I was doing floral biology and immunochemistry. And finally, I decided to continue on immunology after your great classes. So thank you very much.
Thanks.
And regarding the question, so during my PhD and postdoc, I was mainly studying responses in the lymphatic tissues, in the secondary lymphoid organs, in the lymph node and the spleen. And then there was this paper from a Japanese group from the Takahashi lab, where they showed that the memory B cells in the lungs, they were much more cross-reactive than the ones in the lymphatic tissues. And the mechanism behind that is not very clear, but I was really fascinated by this paper in Journal of Experimental Medicine in 2015. And since then, I said, okay, once I will open my lab, I want to study what the B cells are doing in the lungs. And I think this was a journal club I did during my PhD. And then I said, okay, and then when I came here to France, I wanted to study B cells in the lungs. And this is how it started, thanks to this paper that I presented during a journal club.
Well, thank you for the very inspirational words for the new generation of scientists. Now I would like to invite you to share the screen and deliver your lecture.
Yeah, okay. Okay, thanks everyone very much. I will start, perfect. So I will start with a brief summary of the B cell response. We know that when B cells encounter a pathogen, through their B cell receptor, they present antigen-derived peptides to the T cells, which give a first wave of help to the B cells in order to either mount an extrafollicular response, where they will produce a first wave of low affinity antibodies, or B cells can enter into the germinal center reaction, where they will undergo somatic hypermutation, affinity maturation, and diversification of their B cell receptor. So as Gabriel mentioned during my PhD, what I found was that these pathogens are not encountered by the B cells in a soluble way, but are presented by a layer of macrophages that is found in the lymph node. That is, they are called the subcapsular sinus macrophages, and these macrophages play a very important role in the initiation of the B cell response. Furthermore, what I found as a continuation of this study was that this initial T cell help provided to the B cell is not sufficient to induce strong germinal center reactions during infection, but the B cells require soluble factors produced by another population of T cells known as the natural killer T cells. However, when I came to France, I started studying the products of the germinal center reaction, that they are the memory cells. And in the case of the B cells, we have two types of memory cells. We have the long-lived plasma cells, which will continuously produce antibodies throughout the life of the organism, and the class switch memory B cells that they will remain quiescent until the future encounter of a pathogen. And only then these memory B cells can either become antibody-secreting cells, or up to a certain extent, and depending on the model that we are using, they can re-enter the germinal center reaction and further diversify their B cell receptor. So for a long time, we thought that these long-lived plasma cells and memory B cells were mainly residing in the lymphoid organs, in the case of the plasma cells in the bone marrow, and in the case of the memory B cells in the lymph node and in the spleen. However, in the last decade, many groups, including ours, show that these memory B cells and plasma cells can further take up residence in barrier tissues upon infection. And as in the case of the gut, and as in the case of the lung, and this is something that I'm trying to depict here. So how do we study memory responses in the lung? We use a fate mapping system that is an AID-CRE-ERT2 ROSA26YFP. So we infect these mice intranasally with influenza virus, H1N1 PR8 strain. Then between day 6 and 10, we give tamoxifen to the mice. So the cells that express the AID, mainly the cells inside the germinal center reaction, but maybe a pre-germinal center B cells too, they will become yellow. And then we sacrifice the mice after 5 weeks when the virus is gone, and then we check for the YFP positive cells. And in fact, we see that in the lungs, we get a population of YFP positive cells that is a CD45 IV negative. What it means is that when we want to analyze responses in the lung is really heavily irrigated by the blood circulation. So we need to inject 5 minutes before sacrificing the animals a CD45 IV in order to exclude the cells that are in the lung circulation. So we get on these YFP positive cells, and we found that these YFP positive cells are in close association to the bronchi. And we think this is a quite strategic place to be located, because as soon as the influenza comes again through the respiratory airways, the cells will be there all ready to react. We did initially some 2-photon microscopy of these YFP positive cells, and we see that they are really protected by a dense network of collagen fibers. So what are these YFP positive cells? We see that we have still, even after 5 weeks, some ongoing germinal center reactions in the lungs, and we have the memory cells. And inside the memory cells, what we see is that we have around 3/4 of these memory cells are memory B cells, and around 1/4 are long-lived plasma cells. Where are these cells located? So we see that these cells are forming tertiary lymphoid structures, where we can see here staining for BCL6, the germinal center reactions. Then here in cyan, we can find the memory B cells. And then we can see that the plasma cells, they form here in red, they form clusters in the periphery of these tertiary lymphoid structures. So first, I will present you 2 stories. The first one is talking about specifically on memory B cells. That is the work that we published recently from the lab. And then I will show you a second story that we have under revision that is trying to see how these germinal center reactions in the lungs decide whether they will become a memory B cell or a plasma cell. So regarding the memory B cells, this was work performed by Claude Gregoire, engineer in the lab. We performed some single-cell RNA-seq on these lung resident memory B cells. And what we saw was that we found 3 clusters of memory B cells, that they are transcriptionally different. We could separate this cluster because we have this really small one that does not express CCR6, and these 2 big ones that express CCR6. Inside these 2, we can differentiate the green and the red because the green one expresses really high levels of CXCR3, and the red one doesn't express CXCR3 at all. And using these 2 markers, CCR6 and CXCR3, we can identify by flow cytometry these 3 different clusters of memory B cells with similar percentages to the ones observed by single-cell RNA-seq. So what do we know about these memory B cells? First, we did some microscopy to identify as we could differentiate them according to the chemical receptor they were expressing. We thought that they were going to be located in different places, and that's why we performed some microscopy. And as you can see, this small population that is double negative, this one here, is mostly in the periphery of the tertiary lymphoid structures, while the 2 other ones, the CCR6, CXCR3 plus, and the double positive, they are found in the center of the tertiary lymphoid structures. Regarding the isotype that they are expressing, we see that this small population of blue cells, they are mainly IgM, and the other 2 populations, the red and the green, they are IgM, but also class switch towards IgG, and very little to IgA. This was something surprising for us as we are working in the mucosa. We thought we are going to find a lot of IgA memory B cells, but this was not the case, and you will see why after. So I'm not going to talk anymore about this small population because basically what we found was that this double negative population, IgM expressing cells, were present in the mouse even before infection. So they are not triggered by influenza, but these 2 populations only appear after infection. We want to check the mutation low. This is the mutation on the B cell receptor. And what we see is that both populations, the red one that is the single positive, CCR6 plus, and the green one that is the double positive, CCR6, CXCR3 plus. And both of them, they are mutated, suggesting that these cells could be coming from the germinal center reaction. To assess this, we perform an experiment where we give an anti-CD40 ligand that disrupts the germinal center reaction. And basically, what we see is that we lose both populations of memory B cells, suggesting that they are coming from the germinal center. This is not the best way to say whether something is coming from the germinal center reaction or not. Now we have much better tools to do this, but at that point, it was the best we had. But as you will see later, we have now better models. And we wanted to see whether these cells were coming from the same progenitors or not. And for this, we measured the clonal overlap between the memory B cells from the single positive and double positive population. And we found really little clonal overlap between these memory B cell subsets, suggesting that these are coming from different origins. So we went on to study what these cells are. And one of the most striking findings we have on this population was that we found that the double positive population that we call the bona fide memory B cells, they could bind to the antigens of influenza, to the nucleoprotein, and to the agglutinin to a really high extent, while the single positive population, even though they are really abundant, they were not binding to the nucleoprotein or to the agglutinin of flu. At this point, we check whether they could be binding to other proteins of influenza, like neuraminidase or the matrix proteins, but this was not the case. So, and then we thought, okay, we have a population, the bona fide population that is binding the influenza antigens, while the other one is not. So what happens during rechallenge? So one way to see what these cells, if these cells can actively differentiate into plasma cells, is because we lose them after rechallenge. As you can see here, this is the double positive and single positive population. And what we see is that immediately after reinfection, this double positive population goes down really high. And this is because these memory B cells are rapidly differentiating into antibody secreting cells. However, the single positive population still remain there, suggesting that these cells were not differentiating into antibody secreting cells upon rechallenge. Then we wanted to see whether this population, the antigen-specific one, was more protected than this one. So what we did was to do an adoptive transfer of the single positive or the double positive population into naive mice to infect these mice with flu. And what we see is that the mice that receive this bona fide population of memory B cells containing antigen-specific cells, they were mostly protected by the infection, while the ones receiving this population, they were not really well protected from infection. So then at this point, we said, okay, what is this red population, the single positive population doing? Like they are generated, but it seems that they are not antigen specific. So what could which role could be playing during a recall response? So we went back to our RNA-seq data, and what we found is out of the top 5 most differentially expressed genes by the single positive population were the Fc receptors, the Fc receptor for IgE and the Fc receptor for IgM. So basically, we came up with this hypothesis that is that upon every challenge, we will have these bona fide memory B cells that express CXCR3, that they will recognize the influenza virus and rapidly differentiate into antibody secreting cells. On the other side, we will have these CXCR3 negative memory B cells that in our article, we call them the bystander memory B cells due to the lack of a better term, or we can call it in other ways, that this population express really high levels of Fc receptors. So upon rechallenge, they won't be able to bind the pathogen through the B cell receptor because they are not specific, but they could bind the influenza as an immune complex. So this, as these two populations are in the same lung niche, this could somehow increase the density of antigen and present it to the bona fide memory B cells, or they can internalize the antigen as an immune complex to expand, for example, the pool of T cells that could somehow indirectly help the activation of the bona fide memory B cells and overall enhance the antibody production upon a recall. So we did a couple of experiments. I'm going to test this hypothesis. I'm going to show only 2. The first one was to check, in fact, that these CXCR3 negative memory B cells, they were expressing high levels of Fc receptor and that they could bind to the immune complex. And this is indeed the case. When we incubated single positive and double positive memory B cells with an IgM immune complex with the influenza antigen, we see that the single positive could bind much more to these immune complexes than the single positive population. And then we did some complicated mixed bone marrow chimera where we removed the Fc receptor of the IgM from this population, so we could somehow block this pathway. And what we see is that we have a reduction in the production of antigen-specific antibodies during a recall response, suggesting that this is somehow these cells are necessary to enhance the antigen-specific response, even though they are not specific themselves. And this is the first work that came up from my lab here at in Marseille at the CIML. This was a very close collaboration with the lab of Pierre Milpierre. And yeah, so now I will go to some unpublished data that we have under revision, that this is a work done by a postdoc in the lab, Pia Holgado, and she got help from a PhD student in the lab, Lucas Bertoya, for the lung part. And again, using this lung model, using the same mouse model and tamoxifen treatment and influenza infection, as you can see, we get these YFP positive cells that they are completely absent if the mouse is not infected. And we have the germinal center reactions, as I showed you before. But one thing that caught our attention was that in the lung, we have around 3/4 of the memory population of the memory compartment is composed of memory B cells and only 1/4 by plasma cells. So we wanted to see whether this was all. Well, I'm going forward. First, I will characterize these memory B cells. Basically, what we see is that, again, these memory B cells, we have the NP specific and HA specific memory B cells. So they are specific. And then we check the plasma cells. In the case of the plasma cells, the way to check that they are antigen specific is because we do an ELISpot, and we see that they could bind to the nucleoprotein of flu and also some of them to the hemagglutinin of flu. So we wanted to see whether this bias towards the production of memory B cell was also present in other barrier tissues. And then that's why we went on to study this in the gut. So for the gut, we use another infection model that is Salmonella. So we infected the mice orally instead of intranasally with Salmonella. And then we did the same tamoxifen treatment as in the case of the influenza infection. And then we harvest the small intestine, including the lamina propria and the Peyer patches. As you can see in the non-infected animals, in this case, we already have some YFP positive cells in the intestine because this is coming from the response against the microbiota. However, after Salmonella infection, we have an increase, a very big increase, like 3 times more of the number of YFP positive cells in the gut that this is induced by the infection. We want to check on these YFP positive cells. We have some germinal center reactions. We have the memory cells, but what we observed in terms of the memory cells was completely the opposite of what we observe in the lungs. In this case, we see that 80% of these memory cells were plasma cells and only 15% were memory B cells. So in the gut, what we observed was that in the Peyer patches, we have the germinal center reactions and very little memory B cells, while the plasma cells are mainly located in the lamina propria of the intestine. And here you have the quantifications in the Peyer patches where we see the upregulation of the germinal center response compared to the non-immunized mice. And this you can see the plasma cells that are mainly in the lamina propria. Now we can check. We didn't manage to detect in this case for the memory B cells their antigen specificity. We tried several methods, but none of them worked. But we could see that these plasma cells that we see in the lamina propria of the gut, they are specific for the LPS of Salmonella. So we wanted to see whether this preferential production of memory B cells in the lungs and preferential production of plasma cells in the gut was due to the fact that was due to the barrier tissue, or due to the fact that we were using in one case a virus and the other case we were using a bacteria. So what we did is we took the same mouse model and we choose one pathogen. And in this case, it was Klebsiella pneumoniae because Klebsiella can infect both the lung and the gut. So we took a group of mice and we gave Klebsiella intranasally, and to another group, Klebsiella by oral gavage. We did our regular tamoxifen treatment and we sacrificed the mice after 5 weeks. And what we see was that Klebsiella pneumoniae induced mainly a memory B cell response in the lung, while it induces mainly a plasma cell response in the gut, suggesting that this is not related to the pathogen that we are using, but something intrinsic to the barrier tissue. We went on to check the antigen-specific response, and this is, for example, the number of Klebsiella-specific plasma cells of relative to the number of cells in the lung versus the gut. And as you can see that in the gut, we get around 10 times more antigen-specific plasma cells than in the lung. And this is not because the plasma cells in the lung are migrating into the marrow and the ones in the gut are staying in the gut, because when we check the antigen-specific plasma cells in the bone marrow after intranasal or oral gavage, there was no difference in any of the setups observed. So we wanted to confirm that this is, we wanted to know, yes, whether this more production of memory B cells in the lungs or more production of plasma cells in the gut was due to a differential selection inside the germinal center reaction. So to assess this, what we did was to go into the germinal centers and measure the cells that are going to become a memory B cell, because they are called the pre-memory B cells, or the B cells that are going to differentiate into a plasma cells, and these are called the pre-plasma cells. And what we observe in the lung is that we have around 20 times more memory B cells than pre-memory B cells than pre-plasma cells, suggesting that there was already a bias towards the production of memory B cells in the germinal center from the lungs. When we went to measure this in the gut, what we observed first was that we have a big reduction in the pre-MBCs compared to the gut, to the lung, and then we get 2 times more pre-PC compared to the lung. And when we do a ratio of pre-PC versus pre-MBC, we observe that at all time points of infection, we have a favored production of pre-plasma cells in the gut compared to the lung. So a bit to summarize what we are seeing here, we think that the different memory strategies evolve in the lung, in the gut. In the lung, we have a favored production of memory B cells, while in the gut, we have a favored production of plasma cells. So we want to understand why we have these differences between the different tissues, and that's why we went on to study the what could be regulating the selection of memory B cells and plasma cells in the mucosal germinal centers. And what in the germinal center B cells in the mucosas, they need to integrate different informations from their B cell receptor, because, for example, in the case of influenza or salmonella, they will have B cells that have different specificities. It could be, for example, one specific for the agglutinin, the other one specific for the nucleoprotein. They will also have different affinities, because even if they are specific for the same antigen, it could be of low, middle, or high affinity. And also in the case of the mucosa, we also have different isotypes. So we have B cells expressing an IgM, an IgG, and also an IgA B cell receptor. So how do the germinal center B cells in the barrier tissues integrate all this information in order to decide whether it will produce a memory B cell or a plasma cell? So for this, we started using a different transgenic system. The first one was an AID knockout. The AID knockout, they have a diverse BCR repertoire because this is triggered by the RAG proteins and not by AID. But these B cells cannot undergo affinity maturation, and they cannot undergo class switching towards the different isotypes because both processes are initiated by the same enzyme, that is AID. When we infected these mice, wild type or AID knockout with influenza, and then we check in the lung, what we observed was that we have a big reduction in the formation of plasma cells in the lung and a big reduction in the formation of plasma cells in the gut after infection. And this, what I will show you through the rest of the presentation, is a ratio of plasma cells versus memory B cell. And you see that there is a big decrease in the production of plasma cells in both cases. In the case of the gut, I have to disclose that this response is a mix of the response of against the microbiota and also a response against the pathogen. So right now, what we are doing is to measure also by ELISpot the antigen-specific response to show that this reduction is also reflected in terms of antigen specificity. So it means that affinity maturation or class switching may be important for the production of plasma cells in the barrier tissue. So how can we dissociate these two processes? So for this, we got these mice from the lab of Tasuku Honjo, that they have a point mutation on AID. And these mice, they can class switch normally, as you can see here. These are wild-type mice, AID with a point mutation, they can class switch towards IgA. And these are AID knockout that they cannot class switch. But when this group measured the mutation on the B cell receptor, as you can see, the wild type can undergo somatic hypermutation, while the mice with the point mutation on AID cannot undergo mutations, so they cannot increase their affinity to the same extent as AID knockout mice. So then we have a tool to dissociate somehow affinity maturation and a class switch recombination. So we went on to infect these mice, and surprisingly, what we observed was that we don't observe any difference in the production of plasma cells in the lung or in the gut, and the ratio of plasma cells versus memory B cells was the same in both cases. So this result, what is telling us that even if we limit affinity maturation, this doesn't affect the production of plasma cells in the mucosas. And this is quite different to what we observe in the case of immunization, when where with simple antigens like MPK-LH, where the affinity is the main driver of plasma cell differentiation. So we said, okay, if affinity is not driving the plasma cell differentiation, maybe it's different isotypes in the mucosa that is driving this plasma cell phenotype. And ideally, we would have liked to have a system, another mutation that does the opposite, that affects somatic hypermutation. No, the opposite, that affects class switching, but somatic hypermutation is normal. But these mice don't exist. So what we want to check was the isotype distribution on the memory B cells and on the plasma cells. And what we found was that that IgG B cells could enter into the memory B cell compartment or to the plasma cell compartment to the same extent, but the IgA cells were mainly entering into the plasma cell compartment compared to rather than the memory B cells. And as you can see here, here is the quantification of memory B cells and plasma cells on the other all the isotopes, and we see that the main difference comes on the IgA, where the IgA B cells can become mainly plasma cells rather than memory B cells. We check in the gut and we observe a similar phenomenon. We observe that most of the IgA cells were becoming plasma cells rather than memory B cells. So this is something, this is a correlation. So we really wanted to test whether the IgA BCR could be favoring the differentiation into plasma cells. So for this first, we took an IgA knockout mouse. So these mice cannot express the IgA BCR. We infect them with influenza, and we saw that we have a big reduction in the formation of plasma cells compared to the wild-type mice. Then we went to check in the gut, and we also got a similar trend in the reduction of plasma cell responses compared to the wild-type animals. So now we wanted to do the opposite. We wanted to see, okay, what happens if in this case, we see that if we remove the IgA, we have a reduction in the formation of plasma cells. So we wanted to do the opposite. What happens if we take an AID knockout mice that they cannot class switch and they cannot undergo affinity maturation, and then they have a defect in the plasma cell response, as you can see here? What happens if in these mice we force the expression of the IgA B cell receptor? So this is what we did. We crossed these mice with another mouse line where the IgM is replaced by an IgA. And in this case, we will have B cells where they will express an IgA B cell receptor. They won't be able to class switch or to increase their affinity because they are crossed with an AID knockout. And what we observe is that if we force expression of the IgA BCR in an AID knockout background, we are able to recover the plasma cell response to a similar level of the wild type, suggesting that the IgA BCR is sufficient to induce this plasma cell response in the mucosal tissues. So basically what we think is that in the selection in the mucosa highly relies on the isotype of the B cell receptor. So what we think is that when the B cell express an IgM, they mainly become memory B cells. When they express an IgG, they could become plasma cells and memory B cells, and maybe there on the IgG, the affinity could play a role of whether they will become one or the other one. But what we see is that as soon as a germinal center B cell express an IgA B cell receptor, they favor the production of plasma cells. So can this explain the differences we observe in the lung and in the gut? Like what we observe is that IgA can drive more plasma cells, so could be that in the lung we have a lower IgA, and in the gut we have more IgA, and that's why we produce more plasma cells in the gut. So that's why we went on to measure in the germinal center B cells after a Klebsiella infection, both in the lung and in the gut. And in fact, this is what we observe. We see that the germinal centers in the gut, they favor their class switching towards IgA compared to those in the lungs. And here is the quantification. So in fact, what we think is that the fact that the germinal center in the gut, they class switch more towards IgA, is favoring the plasma cell differentiation. So why is this different in the IgA BCR? So one of the main differences between the lung and the gut compartments is that in the gut, we have a lot of microbiota. And then it was shown by the group of Kenya Onda, like more than 10 years ago, that when we have colonization of the gut, with Clostridium, for example, we have a really big increase in the TGF-beta levels and on Tregs. And this is somehow what they propose is that this is somehow to dampen the immune response against the microbiota. But TGF-beta at the same time is not only a regulatory molecule, but also is the main cytokine inducing IgA. So that's why we went on to check TGF-beta expression between germ-free and SPF mice. And in fact, what we see is that the TGF-beta levels in the lungs, there is not much of a difference. But in the gut, we see a really big decrease in the TGF-beta levels in germ-free animals. And then, in fact, when we compare the class switching towards IgA, we don't see much of a difference in the lung, while if there is no microbiota, then we have a big reduction in the IgA production. So now we wanted to have a system where the mice will have like microbiota, but the B cells in the gut, they won't be able to sense the TGF-beta produced in response to the microbiota. So this is what we did. We crossed a CD19 CRE with a TGF-beta receptor flux mice. So these mice, they won't be able to sense the TGF-beta. And as you can see, these mice, they won't be able to class switch towards IgA in the gut. And what we observe now when we infect these mice with Salmonella is that now we have a really big decrease in the formation of plasma cells in the gut. And now we get like a much more memory B cells that is somehow is similar to what we observe in the lung, but now we get this in the gut. So basically, the model that we think how it's working is that in the lung, we have really low levels of microbiota. There are low levels of TGF-beta. We have less number of IgA germinal center B cells, and we have a higher production of memory B cells. On the other side, in the gut, we have high levels of microbiota that this induces high levels of TGF-beta. It favors the class switching towards IgA, and the IgA BCR favors the production of plasma cells. So now in the last part of the talk, I'm going to show what we are trying to understand why the IgA BCR would favor this plasma cell phenotype. And the first idea that came to our mind was the BCR signaling. So in the case, for example, of an IgM, the BCR signal, the initiation of the BCR signaling is strictly dependent on this Ig alpha beta heterodimer that transduces signaling upon antigen recognition. In the case of the IgG, the immunoglobulin itself has a cytoplasmic tail that is completely absent in the case of IgM. And this cytoplasmic tail has conserved motifs with serines and tyrosines that can be phosphorylated and recruit additional molecules that will enhance the BCR signaling upon cross-linking. In the case of IgA, there is a cytoplasmic tail that has serine, it has tyrosines, but we don't know whether it's playing any role on the BCR signaling. So this is what we want to study. The first thing we did was to compare the signaling under the IgG versus the IgA B cell receptor. So this is a work that was done by a PhD student in the lab, Samuel Origlio. We took a germinal center B cells after influenza infection from the lungs, and we stimulate with anti-kappa. So why we stimulate with anti-kappa? Because we didn't want to stimulate with isotype-specific antibodies. Like, and by stimulating through the light chain, we are able to stimulate IgG or IgA cells through regardless of their isotype. And what we observe is that first, the germinal centers, they don't signal a lot, and this was already described. As you can see, the induction of the BCR response after anti-kappa stimulation is not great. But what we observed was that the IgGs were signaling more than the IgS when we stimulate with an anti-kappa antibody. This was the case in the lungs, and this is what we observed in the gut. We observed also there was a small reduction on the IgA BCR signaling compared to the IgG. So this, we were a bit confused because initially we thought, okay, the IgA BCR is going to signal more, and that's why they are going to become more plasma cells. But then we came up with these papers by the group of Kai Telner and Tomohiro Kurosaki, that what they propose is that the BCR signaling is not like what the BCR signaling is an apoptotic signaling. So in the germinal center reaction, the B cells will undergo BCR signaling, and this will induce the accumulation of the reactive oxygen species. But if they receive enough T cell help, then the B cells will be able to survive and then produce plasma cells. What they show these two labs that I just mentioned was that if they increase the BCR signaling in the germinal center reaction by removing phosphatases such as a CSK or SHIP, then this enhances the accumulation of ROS, and the T cell help is not sufficient to recover them from death. And what they see in the system is that they have big amounts of apoptosis and a really big reduction in the formation of plasma cells, suggesting that more VCR signaling doesn't induce more plasma cell, but in terms it's the opposite. So then we thought, okay, maybe in the case of the IgA, we are on this side of the spectrum. As the IgA VCR would signal less, then they will accumulate less ROS, and they are able to survive better even in the absence of a lot of T cell help. So how can we test this theory? How can we modulate the signaling on the IgA VCR without touching the other isotypes? So that's why we want to generate two different transgenic mice. The first one is an IgA TD tomato, where we put a TD tomato after a P2A at the end of the IgA gene. And this we did this mouse in order to be able to track the IgA B cells without using an IgA antibody, because as we are going to study signaling, we wanted to identify them regardless of an antibody. And then we took these mice, and what we did was to remove the cytoplasmic tail region of the IgA BCR. The first thing we checked was the BCR expression level, because we were afraid that removing the cytoplasmic tail was going to change the BCR levels on the membrane. However, we didn't observe much of a difference in terms of the IgA BCR on the membrane, suggesting that removing the cytoplasmic tail is not affecting the surface levels of BCR. And then when we stimulate both the IgA wild type and delta tail B cells with an anti-IgA BCR, what we see is that when we remove the cytoplasmic tail, we have a reduction on the BTK on the BTK phosphorylation compared to the wild type cells, suggesting that somehow the cytoplasmic tail of the IgA is important to for the IgA BCR to signal. And what we found was that if we reduce the BCR signal and the IgA BCR, we also, these cells accumulate less reactive oxygen species compared to the wild-type cells. So then the last question was, okay, now what happens to these mice when we infect them? What happened with the memory response? And what we observed was that when we infect the mice with influenza virus, the wild-type tail and the delta tail mice, and what we see is that when we remove the tail of the IgA BCR, we produce almost the double amount of plasma cells compared to the wild-type cells. At the beginning, this was something confusing, and we thought we were going to get the opposite, but in fact, we get double the amount of plasma cells. We get a reduction in the memory B cells, and when we do a plasma cell versus memory B cell ratio, we got an increase. And this is, as you can see here, this is the influenza-specific IgA titers when we remove the tail region of the IgA BCR. We did the same in the gut, and what we see is that the delta tail mouse, they produce more plasma cells compared to the wild type, less memory B cells, and we also see that the salmonella-specific anti-IgA in the feces is much higher than the wild type mice, suggested again that removing the delta tail favors, removing the tail of the IgA BCR favors their differentiation into antigen-specific plasma cells. So again, what we think is that in the IgA, we are in this zone where the IgA BCR is signaling less, and this somehow allows them to survive better in the germinal center reaction and to produce more plasma cells. So I will finish my talk by making two conclusions or two hypotheses of the impact that this could have. So our first hypothesis is that if the IgA B cells are able to survive and are able to be selected into the plasma cell compartment with a lot of T cell help, what we think is that these B cells won't be of high affinity. And I think this makes sense in the terms, for example, of the gut, where you don't want to have really high affinity B cells, because what we think that in this case, they will be able to produce really high affinity IgA, that this could eliminate the bacteria of the gut. However, what we think is that the fact of having like low to medium affinity of the IgA antibodies in the gut could be helpful to maintain the microbiota there under control without eliminating it. On the other thing was like, why we would like to have different memory strategies in the lung and in the gut. And the way we like to think that is that in the case of the gut, we are exposed to, for example, to bacteria like Shigella, Salmonella, E. coli through poison food or water. And in this case, it's better to have plasma cells already producing antibodies to recognize these bacteria and eliminate it immediately. However, in the case of the lung, we are exposed to these viruses like influenza, SARS, that they are changing and mutating all the time. So we think that in this case, it's important also to have not only the plasma cells producing antibodies to eliminate the same pathogen if we encounter it again, but also to have a big a battery of memory B cells that could be of lower affinity and could recognize the variants of these viruses. So with that, I would like to thank everyone that make this work happen. Everyone in the lab. The work that I presented was done by Claude Gregoire, Samuel, Pia, and the lung part also by Lucas. Then to all our collaborators, in particular the lab of Pierre Milpied that helped us during the first story, to all the platforms of the CIML, and also to all the people that gave us the money, to all the on the French side, but also on the European side, that this was very helpful to perform the studies. And the last slide is just to advertise an animal workshop that we are organizing with all these colleagues here on pathogen immunity and signaling, that will take place in June next year not in Amsterdam, in Leiden, in the Netherlands. So yes, so just spread the word. And thank you very much.
Mauro, for sharing this fascinating work. It's really very exciting. The unpublished data is very, very exciting. So, as usual, this talk will be followed by questions and answers in YouTube. And we invite you to send your questions to all the audience. Thank you very much. And we look forward to meeting you next week with a new global IMMUNOTALK speaker, Dr. Wendy Barclay. So thanks again, Mauro. Thanks for your help and your very exciting work.
Yeah, thank you very much for giving me the opportunity to talk here.